everyone seems to be in now. So we'll start. Xingbo is going to do his third lecture today. Go ahead, Xingbo.
Okay, good. All right, so first I'd like to say thanks a lot for all the great questions so far. I enjoyed a lot. I think this is interaction is the most fun part. So please keep that coming. Therefore, feel free to interrupt me and ask your question as soon as you have one. So, so last time we talked about the measurement of spatial temporal metabolism. So I introduced some imaging-based techniques to measure metabolism in living cells with a single cell and even subcellular or optical resolution. So for today's lecture, I would like to basically present a research project that I did during my postdoctoral studies, where I kind of implemented those techniques to study metabolic control, or more in general, the biophysics of energy metabolism in development. So therefore, I think the purpose of this talk is to showcase that how you actually use those techniques in the research context. So last lecture, I mostly focused on measuring metabolite concentrations, like NADH concentrations, ATP concentrations, or potentials, for example, membrane potentials. On the other hand, metabolic activities are characterized by metabolic fluxes, right, which is the turnover rate of the metabolites. We can measure ATP, ADP concentrations, but the concentrations themselves carry only so much information. Also, what's very important is how fast you are making ATP from ADP, because as soon as ATP is being made, it's being consumed at steady state. So it kind of forms a cycle. In that case, if you measure the ATP concentration in steady state, it's a constant, right? And then if you have a way to measure how fast ATP is being turned over, or metabolic fluxes, that would actually tell you how fast, right, the cells are producing ATP or using ATP, because energy is supplied on the fly. So actually, this quantity is very important. Of course, you can also measure those fluxes through, for example, oxygen consumption rate, which is usually experimentally easier because oxygen comes from outside, or some secretion rates of metabolites that are being secreted from the cells. Okay, so for example, an example of those metabolic fluxes could be this respiration rate, right, which is through mitochondria, how fast it is producing ATP, for example, or glycolysis rate, where also you can characterize it by how fast your ATP is being produced through glycolysis, which is one aspect of this glycolysis flux. So those fluxes, for example, today I'll be focusing more on mitochondrial metabolic flux, as I also focused a bit more in the last lecture about mitochondrial metabolism. So mitochondrial metabolic flux, they change in space and time during development. So what you're looking at here, again, is early mammalian embryo development or pre-implantation embryo development. It starts from an oocyte, right, which I showed multiple times from my last lecture as before fertilization, right? So they are here and matured, they're waiting for sperm. After fertilization, you get a fertilized oocyte or the zygote, and then you would undergo cleavage divisions all the way about day 5. For example, you form what is called a blastocyst, consisting of 100 to 100 cells on that order of magnitude. So if you measure oxygen consumption rate, which is how fast they're consuming oxygen, which is a very important metabolic rate for cells in general, that increases by 3-fold at the blastocyst stage compared to the early stage, for example, the 1-cell stage. That remains to be a mystery. So because the cell volume is not changing, it's basically reductive division. You get more cells, but each cell gets smaller. You don't grow much in size, but oxygen consumption rate increases by 3-fold. And also there's spatial variation where if you look at the blastocyst stage, so that's where the first cell differentiation happens, where you start to differentiate into 2 parts, right? One is called inner cell mass. The other is trophectoderm. The inner cell mass will eventually develop into the embryo. The trophectoderm would become a placenta, right, that supports the embryo. So this is the very first cell division. The cell differentiation. And people have isolated those cells and measured their oxygen consumption rate separately, and they find the trophectoderm has a higher oxygen consumption rate compared to the inner cell mass. Okay. Also, the mechanism behind this also remains unclear. So then basically, we are asking the question, what are the causes and consequences of the spatial temporal variation of metabolic fluxes during development? So that's kind of a big question out there. You can break it down, right? Number one, what controls metabolic fluxes in space and time, right? So what are the processes inside of a cell that controls, let's say, oxygen consumption rate? Okay, how does it change in space and time? Another part is how do these metabolic variations impact development across multiple scales in terms of cell phase specification, cell motility, morphogenesis in general. So those kind of the questions I'm very much interested in. But to tackle these questions, one would need a method to measure metabolic fluxes with sufficient spatial temporal resolution, preferably even subcellular resolution. Right. So for that, so we choose the mature oocyte as the model system because it's at metabolic steady state. So if you measure its oxygen consumption rate, it's a constant over many hours. So therefore, it has a nice baseline. It's convenient for metabolic perturbations around that baseline. You can ask very simple questions like what even controls that steady state flux at this stage, right? So conceptually simple and experimentally also easy to perturb. All right, so okay, then, so any questions so far? So this is just the brief intro about the system. Okay, good. All right, so then, so how do we measure metabolic fluxes? Right, so I already talked about the NADH, right, the electron carrier, which is the electron carrier that donates electron to oxygen, right? So it happens inside of mitochondria, for example, where it donates electron to oxygen through electron transport chain, or ETC, and that free energy is used to pump protons across the mitochondrial membrane, right here, and itself will be oxidized as a consequence into NAD, and then the the proton gradient established will be used to make ATP by through this ATP synthase machinery. And on the other hand, now NAD becomes oxidized, and then you need to complete the cycle, right? You need to go back, become NADH. And there you use the pathways, for example, like Krebs cycle, or you can go through fatty acid oxidation, where you basically use nutrients and that to reduce NAD back to NADH, and then you complete the cycle. So NADH is central to mitochondrial metabolism. Moreover, it's autofluorescent, as I described before. That means you can just image it without any labeling. So it's a very convenient technique that you can just do in any cells. And people have been doing that for quite a while. However, the fluorescence intensity itself alone is not enough to quantify metabolism. I think also we got asked some questions about NADH. Can you quantify them being free or being bound to enzymes? I think you will get your answer today.
Xingbo, can I ask a question? So
yes.
I don't know too much about these electron carriers, but my understanding was that there are in principle more electron carriers in a cell than just NADH, right? For instance, there's also NADPH and sort of other things. I'm sort of wondering, just like what do you have to say about those? And is it generally that we talk about NADH because the flux through it is just so much larger than the other ones? Could you say something about that?
Yeah, exactly. So as you mentioned, there's NADPH. NADPH is more involved in biosynthesis, so it's more relevant in the cytoplasm. And inside of mitochondria, it's NADH is dominating. So NADH is generally more abundant in the mitochondria. Within mitochondria, there's always also FAD or FADH too. which is also kind of, that is pretty much equivalent to NADH, and it's enriched in mitochondria, where also electron carrier. But there also, in general, for example, if you're powered by pyruvate, actually that would depend on which nutrient you use. For example, for pyruvate, which is the case for oocyte, then the NADH is also much more abundant than FAD if you're using this type of nutrients. So therefore, for this study, where I'm focusing on mouse oocytes, NADH is dominating, therefore. So that's kind of the justification. But in general, if you are looking at many other different cells, one has to pay attention to be careful, like which electron carriers is more dominating. And in terms of measurements, NADH and FAD is very separated in spectrum. So you can actually measure FAD in another spectrum, which I'm not going to talk about, but we actually have done already, that give you information about FAD. If you look at this particular spectrum, it's NADH and NADPH. But like I said, NADH is dominating in oocytes, so we talk about NADH here.
Cool, thank you.
Yeah.
Great. Yeah.
Go ahead.
So it's a very fundamental question that the NADH is being studied as a readout for what? As in, it comments up on the mitochondrial activity or metabolism in general, or glycolysis per se, or it's a readout of like what exactly, what process?
Yeah, right. So for example, like you are seeing here, if you zoom into mitochondria, then NADH is readout of the electron transport chain or Krebs cycle, right? For example, and it's more than that. The electron transport chain is dominating basically in terms of oxidizing NADH. So the readout of NADH is in general, if I answer that more generally, it's redox state, right? Because it's reduced and oxidized pairs. It's a redox readout. That's how people call it. Okay, so and so in other words, you can say is how reduced or how oxidized your cell is in a very layman's term. But if you zoom into mitochondria, this redox state is dominated by electron transport chain that is oxidized NADH, and the Krebs cycle or fatty acid oxidation and so on, that is reducing NADH. So it kind of, if you measure NADH, you are probing information of those ETC, TCA, and etc. in the mitochondria. But if you look at NADH outside of mitochondria, and that's basically glycolysis, and as well as, for example, fermentation, which is lactate dehydrogenase, they are the enzymes that are controlling this NADH-NAD ratio and the redox state, therefore.
Okay, so can we say that NADH is a readout for like oxidative phosphorylation, like inside mitochondria when we are looking at it, or when it's outside the mitochondria? Does it more or less levels of NADH can comment on how glycolytic the cell is? Any such like understanding?
Yeah, you could say that. So exactly. For example, here this is OSPHOS, right? The oxidative phosphorylation, which is ADP to go to ATP, and it's definitely coupled to NADH. But the only one caveat is that you also have a proton leak, which I think I briefly mentioned. So the coupling of NADH oxidation to the oxidative phosphorylation, the efficiency is not 100%. So there's always some leak. So that's the caveat. Good. All right. So, and actually we'll get to the leak part, which is actually a very important part, cannot be ignored. So, okay. All right. So then, therefore, to get more metabolic information from just measuring intensity, we actually use a technique called fluorescence lifetime imaging microscopy, or FLIM. Okay, so first I want to just see, like, maybe in the room, how many people have heard of FLIM? Can we do a little survey?
I think 2 people, 3 people, 4 people maybe out of 50.
Okay, all right. So
Xingbo, can you repeat the question?
So how many people have heard of FLIM?
Fluorescence lifetime microscopy.
Fluorescence lifetime imaging microscopy.
Yeah, like of the order of 10%, let's say.
Okay. All right. So I guess it's a very new thing, which, okay, so worth introducing a bit more in detail. So, okay, we know fluorescence, right? Fluorescence is where you shine, light on a fluorophore, you have an illumination photon, you hit your fluorophore, and it will be excited, right? It will go from ground state to excited state. And then after a while, it will decay, right? From the excited state to a lower energy state. And it would re-emit a photon. And then you have your detector to detect that photon. And so that is basically the kind of nature of fluorescence, where in general, like for example, when you're doing like, you can do many epifluorescence, you can do confocal imaging, but a lot of times you are just counting photons, right? So you are illuminating your fluorophore. You can use continuum laser, for example, you can use pulse laser, doesn't matter. But you are basically, depending on how strong you excite your fluorophore, you can excite different number of molecules to excited state, and then they would decay, and then you would emit, you would observe the photons, where, for example, depending on the concentration, the number of molecules being excited, or originally in the cell that are being excited, and your laser power and everything, you would get different amount of photons, and that's intensity, right? So from there, if you kind of control your, let's say, calibrate, control your laser intensity, and you just count the photons, and the photons will be roughly proportional to, related or proportional to how many molecules you have, right? So you can have a readout of abundance of your fluorescent molecules. People can use that even qualitatively just to localize, fluorophores, right? You label your proteins and you see where they are. If you want to be more quantitative, you can count the photons. You can get kind of maybe a potentially concentration of the fluorophores. Okay. However, this technique called the fluorescence lifetime imaging microscopy is one step further. It's the same thing in terms of, right? Like, can you implement a confocal microscope, scanning confocal microscope, or for example, but instead of only counting the number of photons you receive, you actually have a clock. You would keep track of each excitation event where you shine an illumination pulse. So in this case, it has to be a pulsed laser, okay? Because you want to keep track of each excitation event. So each pulse would go into your sample and excite a fluorophore, and then you actually start a clock once that pulse hits your sample, or once the pulse is started, and you have a time, T0 or T pulse. And now the fluorophore will be excited, right? And then it would decay. And then once it decays, you detect a photon and you stop your clock, right? And then you got another time, T photon. And then you actually can compute this time difference. And that's called photon arrival time. It roughly basically gives you this the time it spends, the molecule spends on the excited state before it decays. Okay, it's something related to what is called a fluorescence lifetime. Okay, so you can actually do that for many pulses, right? You have to build up the statistics. It's a stochastic. So you keep shining train of pulses, and those pulses are very, very close, right? They're like 12.5 nanoseconds apart, right? And the pulses are like femtosecond pulses, so they're like very, very focused in time, right? So the really sharp pulses. And then you do that, and for each pulse, you would have a certain probability of excite some molecules, and then the molecules would decay at a certain time. And so then you would just record all that information. You count pulse 1, you count that photon arrival time associated with that pulse, and you keep doing that for millions of times, right? And as you scan through your sample, you would build up a decay curve where the y-axis is all the number of photons, all the pulses, right? How many pulses there are, that means, is the number of photons, because some pulses excite a photon, some will not. So there are all the excited events. For each of the excited events, you would have associated photon arrival time with that. And that would follow a decay curve, right? And this decay curve follows the kind of give you information about the average, fluorescence lifetime, for example. All right, of course, there's more information to that, which I'm going to talk about. And the cool thing is, this is just microscopy. So you do that with each pixel, right? So you scan through each pixel. In contrast to a regular confocal microscope, like I said, you just count photons, so you have an intensity image at each pixel. Here, you actually have a curve at each pixel. And the intensity is nothing but the integrand under this curve. And that's the number of photons, right? So that's what flame is. So it gives you this 4D, okay, let's kind of call it, okay, it's XY, so that's 2D in space, right? And then you have an extra dimension, which is give you this fluorescence lifetime, right? So multi-dimensional imaging data. Okay, is this concept clear?
So,
all right.
I didn't understand what the word lifetime refers to here.
Okay, so lifetime is, okay, it's the time basically between, okay, any molecule, fluorescent molecule have an intrinsic fluorescence lifetime. That means if it's being excited, it will go to a higher energy state, and then it will stay there for a little bit, let's say 2 nanoseconds, and it will decay. Once it decays, you get a photon, right? So how long it stay at the excited state, that's basically the lifetime, right? So if, for example, if you have longer lifetime, means you're gonna be excited and it will decay after 3 nanoseconds. You would get 3 nanoseconds between this excitation and the emission. A short lifetime is instantaneous. For example, the instantaneous is like a mirror, for example. You excite and you just get the photon back. Almost like instantaneously, right? They have the speed of light, but that's just the travel distance. But intrinsically, it's like instantaneous. Pretty almost, right? Of course, there's always time scale. Make sense, right? So if it's instantaneous, you basically get like a delta function here, right? Okay.
Xingbo, I also have a question. So
Yes.
yeah, in these plots that you're showing at the bottom, these scatter plots, I'm just confused about how you can get multiple points for the same time. Is that because you've sort of aggregated over multiple such curves, multiple such measurements?
So this is a scanning confocal. So therefore, I'm scanning, right, through each pixel. So look at my cursor. Can you see my cursor, by the way? Can everybody see my cursor? Okay, like I'm just scanning through, right? So at each point, I can park my beam for a little bit. I can, park here for 1 second. Okay, but in theory, I'm doing this constant scanning, right? So there's a certain pixel dual time here. And when my beam is here, I'm collecting from this pixel. When the beam is here, I collect from that pixel. And the whole scan takes about 30 seconds. So in each pixel, you can do the math. Like you park there on average a certain amount of time. And then that's your decay curve. Of course, they are not imaged simultaneously in that sense, because you are sequentially, right? So you are like scanning.
Okay.
Yeah. Okay. Cool. All right. So now,
there's another question, Xingbo.
Yes.
Mike, yeah, okay, it's working. So I wanted to understand the similarity with a common epifluorescence microscope that we generally use in biology. So how would the lifetime vary with respect to an epifluorescence microscope? Does the bleaching occur faster for a fluorophore in case of epifluorescence, and here it does not? Is that the difference, or I just didn't catch that?
Yeah, very good question. So epifluorescence. So this is okay. The distinction should be made between epifluorescence and confocal microscope. This is basically a confocal microscope with this additional clock. That's it, right? So it's basically a confocal microscope. So a confocal microscope is where you, in this case also, it's a 2-photon confocal microscope. So where you have you have light focused very, very well in a point. So you have so-called point spread function, which is like, focused. You can do very nice, let's say, optical sectioning. So if you illuminate your sample 3D, for example, but you can actually just focus on one plane, right? And only the everything within really this point spread function, which is very, very narrow, both x, y, and z, that can be excited efficiently. You don't have much of fluorescence from outside, above or below this focal plane. So you can do very, very nice cross sections. That's what confocal is about. I mean, epifluorescence, I think, illuminated basically. Okay, I'm not a microscopist, so if I'm said something wrong, people should correct me here, right? So epifluorescence, in my understanding, is they basically illuminates, you can get photons from many of, across the light paths. So you don't actually focus on one plane. You actually get information from pretty much averaged across all the light paths. So that's one possibility. And does that answer your question?
Yes, I got that point. And a follow-up question was that, does this microscopy also have any specificity with respect to the region of the cell it's helping to visualize, as in the cell membrane or inside the cell? Because if we compare it with a turf microscopy, which is a total internal reflection microscopy, that is specialized for the membrane. So if we are trying to study something, towards the membrane or some kind of vesicle that fuses out of the membrane, so that helps us. So here also, is there any, spatial difference, a spatial specification with respect to which the microscopy can focus and give us details on?
Yeah, very good question. So let's go back to this kind of confocal, right? Confocal microscope, which you can do very nice optical sectioning. So when I say optical sectioning, I'm not sure if you heard like the Z-stack. People call it Z-stack, right? So it's basically if you have like a cell, I will actually show but it's a very good question. It would help understand the image I'm going to show you. For example, our oocyte is like a 3D sphere, right? When I image it, I can literally do cross section. So I want to see what's here along the equator, or want to see close to the membrane down there, I can do that. Or if I want to see the top, I can even do the top. That just means I just need to focus my lights, my focal planes on different, Z positions along this direction. Of course, usually the signal is much better, closer to the bottom, because the light travels more, you can absorb even more. But the cool thing about the confocal is like they actually can have very deep penetration. So you can easily image, let's say, into 100 microns, no problem. And you can literally see the whole 3D structures, one slice at a time. And I'm actually going to show you some of the slices. Good. All right, very good questions. All right, so, okay, and then why do we measure? Why do we care about fluorescence lifetime, for example, for NADH? It's because NADH have two different forms, right? One is free form. So in general, I'm using the term form loosely. So it can be free, right? It's just freely diffusing, or it can engage or be bound to a protein or enzyme. It has to be bound to a protein because basically it's a cofactor, right? So it binds to a protein or enzyme, and then it can participate in the redox reactions, and where you can start to transfer electrons to its acceptors. So this is kind of the first step. You have to bind, and then you react. So therefore, the cool thing is NADH free and NADH bound have very different lifetimes. So when they are short, they have a fluorescence lifetime of 0.4 nanosecond. That means you shine a pulse, you're gonna expect on average to get a photon after 0.4 nanoseconds. But when they are bound, the conformation changes. So I don't know too much about the photophysics details, but empirically, we know that if they're bound, the lifetime increased to, for example, 2-3 nanoseconds. Depends on exactly what kind of enzyme they bind. They might have different variations, but it's many times more, right? So now if I bind and you shine a pulse, you expect to see another photon after 2-3 nanoseconds. Okay, so that means now if you remember that decay curve, then they would have like fingerprints of those information. So if it's all free NADH, you would have a pretty short lifetime, so it decays pretty fast. If you have all bound NADH, have a longer lifetime, very different curves now. If you have a mixture at steady state, you would have a certain fraction of free, certain fraction of bound. Okay, so I'm going to say NADH free, NADH bound using the following notations. Okay, here basically is the image, actual microscope image. So I think this is a cross section now. So it's basically a confocal image across the middle of the oocyte, right? So right in the middle, where you actually if you will, the radius is the biggest. If you move up and down, you can see the shrink, basically. So I'm basically cutting optically through the middle, and I can see everything inside of the cell, right? So here you see the intensity image. So if you do a confocal microscope, this is what you get, right? And you basically can count the photons of each of the pixels, and I'll give you the contrast. And the brighter ones have more photons. The pixels have more photons, and those are mitochondria. Number one, because we validated it, we just labeled mitochondria with another marker that just labels mitochondria fluorescently, and they just co-localize with these bright pixels. This is NADH image, right? So, but also this is consistent with the fact that NADH is enriched in mitochondria. So that's why it's brighter. Okay, so those are mitochondrial pixels. The rest are cytoplasm. Okay, so like I said, so NADH is involved in oxidative phosphorylation in mitochondria and presumably glycolysis outside. But oocytes, as we know, as people have shown, that it's not much of glycolysis. So we'll be focusing on actually mitochondria today. But in addition, you have this decay curve, right? So this decay curve, I basically just averaged over the whole image, and that's kind of the decay curve of all the NADH molecules, that excited through in this cross section. So that gives you kind of an average information of this, of this plane of the oocyte. Indeed, you have this fast decay component, and slow decay component, right? This is a semi-logarithmic plot. If it is only one decay time scale, you should see just linear, one slope. But apparently you have more than one slope. And minimally, you can consider it as two slopes: fast decay, and slow decay. And the fast is corresponds to free NADH, and slow corresponds to enzyme-bound NADH. We know this because, for example, myself, I did experiments where I just mix some enzymes with NADH in a solution, and I would get the similar curves here. And using this curve, I can actually back out how much NADH is bound and free. And the way I do it is I just feed this curve to a double exponential function, right? So I approximate this as a two-component, system where I have a free NADH. So that is have a short lifetime here, and based on the photophysics, I know it's a Poissonian process. So you put exponential here, and then you put a certain fraction of that component, and of course you mix that with a bound population, and that's your minimum model. You can fit to this curve, and it fits pretty well to the data, number one. And then if you feed that in a solution, you can really recover the correct amount of bound and free NADH that you put in. So it's pretty quantitative. And then in this case, you can get the information that's relevant for our subsequent discussions is the intensity, which is basically, like I said, the integrand under this curve. That is related or proportional to the concentration of NADH if you fix your laser power, for example. And the amplitude is amplitudes are just a fraction of the free and the bound NADH components, right? And they are defined to be vary between 0 and 1, right? So let's say, for example, if it depends on your system, you can get like 40% of free NADH, 60% bound NADH. For example, you can fit this quantitatively, and those are very important metabolic parameters. Okay, so this is additional information that you cannot get just from intensity imaging. Okay, so but why do we care this kind of free and bound? Oh, okay, well, let me first.
I have a question. Can I?
Okay, go ahead.
Yeah. So there must also be a triplet state component on this graph, right? So where would that lie?
The what? Sorry.
The triplet de-excitation, triplet state. So some photons would arrive very late, right?
Okay, that's a good question. So maybe that will translate into more than two components, right? For that, that's why I was saying this is approximated as 2 exponential, but in reality, there could be multi exponentials, and also depends on the resolution of the fluorescence lifetime microscope, which the longest lifetime component you can resolve is bounded by the distance between the pulses, right? So you shine one pulse here, and then you record decay, and then you follow up with another pulse. So you can only measure maximum the distance, the time distance in between. That's about 12.5 nanoseconds. So things longer than that, it's hard to measure, right? So, but if it's within that, you can presumably fit more than 2 exponential to this system, but you would probably risk overfitting, and also it would depend on how separated your components are, right? Like we know free and bound are well separated. If you have another state, which is probably also very much well separated from these two, I think you can reasonably resolve that. Some people have tried to fit more than double exponential, but in here I think I'm focusing on double exponential because both in vitrally I have verified that I can very nicely get the free and bound components. So you can consider this as a kind of a coarse grain measurements. I measure free and measure everything that's not free.
Thank you.
Okay. Cool. So now.
So
yeah.
let's say NADH is bound to different kinds of enzymes.
Yeah.
So is there a way to differentiate?
Yeah, it's a very, very good question. Again, I think it's going to be similar. If there's two different enzymes, when you bind, you have very drastically different lifetimes, then I think you can probably distinguish them. People have done those in vitro experiments where they mix NADH with the lactate dehydrogenase, with malate dehydrogenase. I think, for example, they probably induce a change of, for rest lifetime, let's say, I don't know, from 2 nanosecond to 3 nanosecond. And that's usually, it's not very easy to distinguish. Of course, the more photon you have, the better. It can help you presumably distinguish that. But I would say it's possible. Again, depends on how separated they are, and depends on your signal noise ratio.
Thank you, sir.
Yeah. Okay, so here we also do this segmentation. So like I think people also ask NADH in mitochondria and cytoplasm. Yes, they are in both compartments. So therefore, we don't want to mix them, right? So you want to segment the pixels in the first place. So well, one thing you can do, you can just put some mitochondrial dye, right? And then you can just use that as a mask and to give you mitochondrial pixels, and then you only look at all the signal of NADH coming from those pixels. Or you can just use NADH themselves to do a segmentation because they also create a decent contrast for that matter, which is what we did here. And then you get mitochondrial pixels. They're just gonna give you a mask. And for now on, I'm just gonna only integrate the signals within those pixels. So therefore, I'm studying mitochondria. I'm not contaminated by cytoplasm signals, but you can do the reverse as well. So now the thing is, okay, all right, so I introduced that we can measure NADH intensities, bound and free components, but are they good readout even of metabolic perturbations? So then let's try that, right? So let's test this. So we can do quantitative perturbation of mitochondrial metabolism. Again, I go back to this oxygen drop experiments that just keeps coming up because I think this is a very cool experiment where you can, oxygen is a substrate, right? So you can actually tune oxygen level here again, drop it and recover it. Now let's look at the NADH. I think you already see this, but it's just more in detail. So I'm dropping oxygen and this happens. And this is recovering oxygen. So one more time, I'm dropping. And when it's very, very low, okay, this happens, and recover, this happens, right? So if you look in closely, you can see the mitochondrial pixel sort of brightens up, and then it recovers. So I think people also ask me, right, like how to interpret this. And now let's talk about it in more detail now. Okay, so what does this increased intensity actually even mean? So we can quantify that, right? So let's integrate the intensity quantitatively, and we also measure the oxygen levels. In this case, the oxygen level is in the environment, in the chamber. That's where the our OSS lives in. So environmental oxygen level, and NADH intensity is integrated from mitochondria. So you see, actually, I think someone asked the question also, like when did they start to respond as a function of oxygen? You can actually see it don't respond immediately when I drop oxygen. I drop oxygen, it's like not much of a change, and then below a certain level of oxygen, then has started to increase. The NADH intensity starts to increase. So it's not linear, right, in this case. And then I have another information, which is NADH bound ratio. So this is basically NADH bound over NADH free. Okay, so let's remember that quantity. So this quantity also decreases, right? So that means as you drop oxygen, you have relatively less bound and more free. And you can calibrate this intensities, and then you can make that into actual concentrations. And if you do that, you can actually see that, yes, indeed, the NADH-free concentration, right, is responding, and in the way that it actually increases, it's more free NADH at very low oxygen. NADH bound is more or less constant, okay, and that results in a decrease of the NADH bound ratio. So that's what happens. So now you can also think in terms, if you want, you can think in terms of the free and bound NADH concentrations now, right? You can even think in terms of absolute concentrations in the order of tens of micromolar, right? So those are the numbers, right? Or actually dozens of micromolar. So then the question is, how are the free measurements related to mitochondrial metabolic fluxes? Again, because I just started by saying metabolic activities are quantified by or characterized by metabolic fluxes, but now I'm just measuring concentrations. I'm measuring more concentrations and measure free and bound concentrations, but how are they related to fluxes, right? So go back to our original questions. So that's kind of a challenge, right? We need a model of mitochondrial metabolism, right? We need to know how the NADH are actually engaging with all the enzymes, and so on and so forth.
I have a question here.
Yeah.
So I'm just trying to understand the last graph. So the NADH bound and free form. So I was thinking, shouldn't they be complementary to each other? As in, if the free form falls, then shouldn't we also expect a similar trend in increase in the bound form with increase in oxygen? Because if it's not free, then what is the other state that it is going to, apart from being bound, that the graph is not reciprocating the two trends?
Very good question. There's NAD. There is a oxidized form, right? So therefore NADH itself is not conserved. It can go into the oxidized state, and then you don't see them, and then they lose fluorescence. So therefore, exactly. So you don't necessarily need to expect to see a reciprocal or this conservation of this. So, and this presumably is what is happening, where when you are dropping oxygen, they cannot be oxidized. So they accumulate in the reduced state, in the NADH state. So NADH global concentration increase. But if we were to measure the NAD, which is the counterparts, I'm going to show a diagram soon. And then here I would predict the NAD, if you can measure concentration, that would be basically complementary to this plot. Okay. So, okay, actually, I'm going to just show that now, right? So let's go back to those cycles, right? So that's why I always remind people to think about, right, like the cycles and what are the conserved quantities, for example. So mitochondrial metabolism, again, it takes in pyruvates. In this case, for oocytes, it definitely takes in pyruvates, as one of the major substrates. Yes, another question?
Yeah, I had a question from the previous slide.
Yeah.
Yeah, so here with the drop in oxygen concentration, the amount of free NADH, that's when we see an increase. Do you think reverse electron transfer has any role to play in this?
Reverse electron, a very good question. Yes, electron transport chain are known to be able to even reverse. Did they reverse here? Okay, good question. I would say, okay, I have to say okay, I think I cannot rule that out, probably, but yeah.
Yeah, I'm just trying to understand if it's just like the NADH is being formed from the catabolic processes and accumulating, or are the catabolic processes being shut down, but as a in order to not let the membrane potential be built up, it's the reverse electron transfer that's contributing to the increase in free NADH.
Very good question. Yes. I would say, yeah, I have to say, actually, I do not know the mechanism of why free is accumulating. Like I said, the more I can speculate is that as a whole, NADH is probably increasing. I can understand that. It's presumably the minimum, hypothesis I can say is because you shut off electron transport chain. Let's pretend you are not changing. Okay, let's pretend first you're not changing much of anything else, right, including the catabolism, as you mentioned. And then basically you're shutting down the oxidation, and then the other parts wings, right? And it makes more NADH. And so NADH level increases. But why it increases in this particular fashion, that the free increases, but less than bound, more than bound, increase more than bound. That I do not know. But would that necessarily involve the reversal of electron transport chain? That I'm not sure, but I cannot rule that out, I think, at this point.
Okay.
But I will try to talk about the metabolic fluxes. So I think the fluxes, that's why it is also interesting to look at fluxes, right? So fluxes would tell you, okay, if it's positive, right, then that's easy. But in the following a certain convention, it's in one direction, it's negative, it's the other, for example, right? So I think maybe you will learn something from that. So let's see if we can, say something about this.
Sure. Okay.
Yeah. Okay, so mitochondrial metabolism, again, look inside. That's where the cycles are, right? So, okay, again, so the previous question, we actually have NAD, right? And that we are not measuring. Okay, so okay, so you have NADH cycle, you have proton, again, a proton cycle, you have ATP cycle, okay, 3 couple cycles. But right now, we're just looking at NADH, right? The first cycle, if you will. And then for NADH cycles that we can actually zoom in to account for the actual enzymes, right? So therefore, because we're measuring, the enzyme engaging NADH and free NADH, so let's model those. So you can actually start with a very generic picture, right? So the enzymes can be categorized into oxidases. That is basically in oxidizing NADH to NAD. And the other category is reductases that basically reduce NAD to NADH. It's more of a conceptual separation, right? Because at a cycle, you will have some enzymes primarily running in one direction, oxidize NAD, and the others is reducing NADH. So conceptually, you can separate them. And there can be many enzymes doing that. So therefore, we don't go to a very constrain ourselves from a more general modeling perspective. So we account for all possible enzymes and the reactions. We also don't restrain ourselves. They can be of arbitrary order with arbitrary nonlinearity. So it sounds very, very general, right? But what can we do with those type of general model? Well, we coarse grain it, right? So that's another concept of coarse graining, where I think this is also related to the question somebody asked, like, can I distinguish the different bound states? That will be challenging, right? So therefore, I would first just to group the enzymes, such that at least I can distinguish free and bound. So therefore, I group them also according to whether they're oxidizing or reducing. So I group everything into oxidase, just effective enzyme, or effective reductase. And then there's a coarse grain model right here. Basically, you can do this mathematically, rigorously, and then you can actually redefine all those kinetic constants or kinetic rates, parameters of the models rigorously. That will look very, very complicated, but that doesn't matter, because the next step is we assume we're at steady state, which we are. And in this case, the steady state really is relative, right? So the steady state means it's steady state relative to the rate of change of, let's say, the concentrations is much slower than those intrinsic kinetic rates, right? By which I mean the kinetic rates, for example, are really fast, though they're usually like seconds, right? So this kind of binding rates or even the reaction rates, they're like seconds, right? Per molecule, for example. And but if you image NADH over time, in this oocytes are just flat. So that's actually pretty much a bona fide steady state. Of course, if you image it for a day, it will probably start to decrease or increase or change. But that separation times of time scale means we're at a quasi-steady state, right, for our reaction networks. So if we are at steady state, we can even further coarse grain. That means we can basically describe everything as an oxidation rate and a reduction rate. That basically means all the fluxes is just one flux here, right? Any of those steps are balanced by any other steps, right? So everything is in steady state. That's why I can do this simplification. Okay, but why do I do this simplification? I'm not saying I'm neglecting all the bound states. That's very important, because here, if I'm looking at now the global fluxes now, which is in this case, if you look at this branch, it's just oxidative flux, and that is electron transport chain, right? Because that's the reactions that we know from biochemistry perspective is doing the oxidation. And that rate is nothing but this concentration of NADH times the effective, very, very coarse grain oxidation rate. This is basically by definition. But this effective oxidation rate, that actually contains all the coarse grain information. So that actually through our model at steady state is predicted to be related, to be proportional to this bound NADH over free, this quantity we call NADH bound ratio, which actually showed you the plot about this bound ratio minus this same quantity bound ratio, but at equilibrium. Okay, equilibrium means you have zero flux, right? That's by definition. You have detailed balance. All the reverse and forward reactions balance each other, so you have no net flux. And that's consistent with this, because if you are at equilibrium, this quantity is also the same as this, and you have zero flux. All right, so these are our model predictions where we related basically concentrations to flux, right, in terms of the bound and free. Yes.
So the equation that you are describing right now, is that a phenomenological equation, or is this a derivation out of your other assumptions?
It is a derivation. It's an explicit derivation following the assumptions of steady state, where you do the coarse graining. So therefore, there is a proportionality constant, which would actually be quite complicated as a function of some of the rates in the model. For now, we assume that is a constant, and let's see how good that assumption is. And then this equilibrium parameter would also be pretty complicated as a function of some of the model parameters. But luckily, I will show you that we can measure this. So therefore, this is a very general relation. Doesn't depend on the details of the detailed exact kinetic model. It would only depend on the steady state assumption. Of course, detailed model would modify a little bit of those prefactor, the form of the prefactor, which is a prefactor.
Okay, thank you.
Yeah. So that's why I think that's why it's kind of a generic coarse grain model. And therefore, in that sense, I think it's kind of a power of coarse graining, which I also talked about in the active matter parts, right? Some of the form of the equation is pretty much very generic. It only depends on symmetrical arguments in the context of active matter. Detailed kinetic models modify some of the physiological parameters, right? And here, so you can even write this down from a certain type of, if you will, you can actually do it from a very, very generic thermodynamic arguments, and you can put an effective parameter in front. And this coarse graining just help you explicitly give values or give functional forms of the physiological parameters as a function of the detailed model. Same thing here.
I have a question too. So I don't know if it makes sense, but if we are considering the protons buffering, like there is a lot of Ross components into the mitochondria. So I was wondering, when you raise the level of oxygen, could it change the equilibrium state of NADH in the sense that maybe the balance between NADH and NAD will be, you can follow this with your techniques, but then maybe there will be some favorized oxidant and reductor towards the choice of NADA and NADH. I don't know if it makes sense, but what could be the balance between the different redox couple, and what could be the consequence on the interpretation of protons flux? Yeah, very good. So that's actually what I'm trying to show in a sense, but okay, to get to that question directly, yes, for example, when you are those other redox pairs or even reactive oxygen species, they can potentially also proton forces that potentially impact the redox state of this, NADH, and that's precisely what we're reading out, right? So we are literally measuring this right now. So let me show you this slide. What I mean is we are right now measuring NADH, right? So we are just reading it off. Okay, there could be many things that change this NADH, but we measure it, right? So you can measure this with flame, you can measure that with flame, you can measure this also with flame at equilibrium, where when you crank up your
crank down your oxygen to, as low as possible. In this case, we literally crank it down to almost zero, and then that's your equilibrium. Okay, so all those things can be measured, right? So when you do the oxygen drop, they are all changed. Like, for example, okay, these are like all, the NADH bound and free, they're all changing, right? So, but why do they change? Are they changed because of some reactive oxygen species, because of proton motive force, or because something else? Mechanistically, I do not know, but that doesn't matter, Then therefore, I'm doing a measurement as a readout of NADH as a consequence of everything that can change it. And in a way, the model, right, that I proposed there, even though I did not explicitly model the other processes like the reactive oxygen species or some other redox pairs, but they can be implicit in the model parameters, right, in the kinetic rates. So that's why I say it's complicated. So therefore, I'm not limiting myself just to say, therefore, I'm not constraining myself in other way, like what can change NADH or not. I'm just reading it off. Does that make sense?
Because so you are measuring some ratio. And I was wondering, yeah, you know that there is other component that can balance the proton flux, but imagine that you interpret the evolution of a ratio, but this is not based on discrete events that could happen. And for example, if you don't consider the NADH synthesis as molecules and not as redox couple, then you could not be able to know if there is more of molecules while there is in the same moment that there is a balance ratio that is changing.
But I would say I'm measuring both. I mean, this is just a concentration. I'm measuring NADH concentration as well. So if the global level changes, that will be accounted for as well, right? So if there's some more synthesis, it will impact, presumably, the absolute concentrations. At the same time, I'm measuring the ratio, which can be thought of as a potential. So I'm measuring both the potential and the concentrations, and they can be affected by many other redox pairs as well as synthesis, right? So that's all still consistent. And actually, this equation now finally related everything together. It related absolute concentrations, it related redox potentials, and it related turnover rates, and it related it to flux. So that's why it sounds quite, it is generic, right? So it is very generic. And you can, if you want, you can go through maths to show that, to convince yourself that if you start with all the things you have, and now you put some of the things you cannot measure into effective rates, it will become this form that will come out. The only assumption is steady state assumption. But of course, maybe I think I would say, I have to admit that maybe, I'm open for people to think about this. Maybe there are some things that we missed that would render this form different. But so far, I don't realize those. And the things you talked about, I think would be interesting to maybe keep thinking a bit more about. Okay, good. So now I think the message.
I had a follow-up question. Sorry.
Yeah.
So the equilibrium ratio of bound and free, this ratio, the equilibrium that you are defining at 0 oxygen level, would that not depend on oxygen level at all? Because the equilibrium, I'm guessing, would depend on what is the external oxygen concentration you have, right? Is there any particular reason you are taking the 0 oxygen level to be the equilibrium value here?
Yeah, that's a very good question. So that's assumption. Yes. So I'm assuming, so first, okay, equilibrium NADH. So equilibrium is defined as it's a particular state where the net flux is 0. That's what equilibrium means, right? It's detailed balance. So therefore, the flux I'm talking about here is net flux, right? So therefore, by definition, there is only one equilibrium flux, which is 0, right? So that basically means if you want to get to equilibrium, you have to suppress completely your electron transport chain. You can do it by dropping oxygen, by inhibiting enzymes, but whatever you do, you would make sure your flux is 0. So therefore, when I drop oxygen to 0, you can measure your oxygen consumption rate, it will become 0. So that's equilibrium. And then I read off a NADH bound over free at that point, and I call it equilibrium, NADH bound over free. But you can maybe ask this question, right? Also, it's a related question that you asked, is that maybe it depends on how I treat my cell. You know, even though at equilibrium, the net flux is 0, but this bound over free, you know, ratio probably can take on multiple different values depending on how I get my cell to equilibrium. That I have tested by doing the following, that I actually get my cell to equilibrium by both dropping oxygen as well as pharmaceutically inhibiting complex 1. And that gives me the same value. So I take that as a kind of a, you know, test of that hypothesis. Okay, so let's.
I'm sorry, just finishing it off. There are no subtle differences here between your equilibrium and your steady state here, right?
No,
because like if you cut off the oxygen level, you are going towards an equilibrium. But I'm just wondering if you are looking at steady state in general, would there be any difference, subtle difference here?
Yeah, that's the whole point actually. If I am not at equilibrium, then these two parameters are different. So I would show you that. So by which I mean is like, first I drop oxygen to 0, and then there I say this is my equilibrium state because oxygen consumption rate is 0, and I read off a quantity called NADH bound over free. Okay, and that's one number. Okay, and now I crank up oxygen to whatever value I want, and then that's a non-equilibrium steady state now, because now they actually have non-zero oxygen consumption rate, and there I measure my oxygen, I measure my bound over free, and that is this generic quantity right here, which I do not have the equilibrium right here. So that's the non-equilibrium steady state value. And how far you are from equilibrium tells you, you know, in this case, the model predicts is related to, you know, the turnover rate of NADH.
Yeah, thanks.
Yeah. So I'm actually testing this now, right? So this is, so at the end of the day, or this is probably also another message is, of course, you make all the assumptions. The one goal, you know, only one way to find out, right, is you actually test it, meaning you say you made a prediction. Okay, this is my prediction right now. So this is the predicted flux through electron transport chain. And also, I normalize it because I do not know exact value of the prefactor. So I set it to, I just normalize it. I just assume it's a constant. So for that, I just predict based on a measured bound over free at all those non-equilibrium steady states, and one measurement at equilibrium, I predict this is how oxygen consumption rate, or sorry, how the electron transport chain flux will behave as a function of oxygen. Only one way to find out is like you have to measure electron transport chain flux directly, right? And the electron transport chain flux is just oxygen consumption rate, right? Okay, neglecting any non-mitochondrial related oxygen consumption rate. Okay, so making that assumption. And then we are now measuring oxygen consumption rate. So this, I think, in my last lecture, I briefly showed this picture, but I did not explain this because I think, you know, other instructors, I hope Jonathan probably already have already talked about oxygen consumption rate. This is another particular, you know, oxygen consumption rate measurement device that just uses a very interesting principle that it converts oxygen consumption rate into spatial gradient of oxygen you can measure. The mechanism is very, very simple. You have your oocytes at the bottom of the well. It's sealed from all sides except the top. Okay, so then you have oxygen diffusing from the atmosphere into your well, and then it's been consumed here. So from reaction diffusion at steady state, because it's a very, very thin capillary, you will have a linear gradient established, and which we did see that once we do the measurements. And then your oxygen consumption rate is just can, you know, through the flux balance, can just be written as the diffusive flux of oxygen, which is the diffusivity times the cross-sectional area of this device, of the well, times the gradient of oxygen that you can actually measure throughout this well using a very sensitive oxygen microsensor. So, I mean, this is actually a commercially available system that you know, I think is very useful to study like this small embryos. Okay, so small embryos and oocytes, for example. And then if we do that, here's the measurements. So we do the same oxygen sweep experiments. We measure oxygen consumption rates with the same on the same sample, but directly now. So this is how to test the theory. And there's no fitting parameter. So I would say this gives us a lot of confidence that our model and model assumptions are reasonable. Okay, so any questions here? Everybody's convinced? I'm surprised. Okay, all right, so now, okay, so with that, I think basically we showed a method, right, that, you know, can be used to measure metabolic flux, and this is mitochondrial metabolic flux or electron transport chain flux with single cell or even subset resolution. Okay, so through imaging, which is actually very powerful technique. So with that, let me see how much time do I still have. Okay, so so with the rest of the time, I want to kind of use now this model and this the frame to answer some fundamental you know, metabolism questions, right? So number one is what controls mitochondrial metabolic flux in oocytes? Like I said, or what sets the oxygen consumption rate of the oocytes? So like I said, even the simple question that your oocytes is at a steady state, right? If you measure oxygen consumption rate, if you don't do any perturbations, you have a value. But what even sets that single value? So I think this is a very general question that you can have many more complicated versions of that. For example, when you develop, your oxygen consumption rate is changing, and what contributes to that? But I think as a very first step, let's address that in a steady state oocyte. So so now, okay, so here I'm again showing you this type of coarse grained view of metabolism. You can see that so far we talk a lot about NADH cycles, proton cycles. We can even measure about ATP, ADP. Well, here is a one arrow that actually includes many things, right? That's whatever is converting ATP back to ADP, right? That's many things. That's a lot of things in the cell, including protein synthesis, right? That they use ATP. Ion pumping, there are a lot of ATPases. Cytoskeleton assembly, you know, the polymerization, motor activities, they all use ATP. And there are many, many things that are using ATP. So we classify this as demand, right? So, you know, or like energy demand, right? I think you'll hear these words a lot, but what do they even mean? What is energy demand? In this sense, I think it's basically imposed by all the ATP users, you know, tap into this ATP hydrolysis rate. So that we can define that as demand. On the other side, right, what powers this whole chain of reactions is supply, right? Eventually you need nutrients from outside. And that could be nutrients, right? And then it would have to run a lot of those, you know, enzymes that, for example, reduce NADH, right? So, and the supply can be many things. It can be sugar, it can be fatty acids, it can be internal stores, which fatty acids is usually, you know, important internal stores, and amino acids, for example. Okay, so, but this kind of course grained view. Now, we want to just see, okay, so the hypothesis is probably demand that's driving the oxygen consumption rate, or maybe it's supply. So then we actually go back to this model where we say, now we can measure oxygen consumption rate now, right? So we basically say, oh, we can measure oxygen consumption rate by measuring the concentration of NADH and as well as both bound and free. And then we can just use this simple equation to predict that. Of course, the bound and free also go into the turnover rate. So, but here I'm going a little bit, so in order to represent the data comprehensively, I can break down the flux into two pieces, right? One piece is this turnover rate, which actually have that in the form of bound over free minus bound over free at equilibrium. So that's your turnover rate of NADH. That's effectively how fast you are oxidizing one single NADH molecule. And of course, the flux is related to also how many molecules you have. So therefore, you have concentration, multiplied, with the turnover rate. And that I put it on x-axis. So therefore, this basically parameterized metabolic flux. And here I'm going to just put oocytes, and then I'm going to perturb around it. Okay, so this is your control condition. Now I can perturb the supply, right? So if you want to know if supply is changing, is controlling the metabolic rate, you just change your supply. And by that, you can change your nutrient conditions. So you can change your pyruvate by 2 orders of magnitude here. Or, like I said, you can also just, some of the enzymes are involved in reducing NADH, that's also supply, just inhibit them or modulate them. And so there's some pharmaceutical perturbations you can do. So through those perturbations, you can actually very significantly change the concentration of NADH, right? If you image, you can see NADH going dark and bright. And then you can actually, using the model, predict that their turnover rate is changing as well. Okay, so that's why they're spread across this XY axis. All right, so however, okay, here's a interesting observation. They're not all over the place though. They are actually along a straight line, within a margin of error. But what does this line means? Okay, this line is where the flux is constant, right? The flux is a constant, meaning because if flux is a constant, right, let's say Jox equals Rox times NADH free equals C. And now you can plot Rox equals C divided by NADH free. So that means these 2 quantities would just be a linear. The relation between this quantity would be linear if you have constant flux, which is what's being observed. So therefore, that's why I conclude that perturbing supply impacts your NADH turnover rate and your concentrations of NADH, but not the flux. So in other words, the concentration and turnover rate changes in compensatory fashions to maintain the flux. So I think it's a pretty non-trivial observation, but of course you have to test it because it's just from a model prediction. So we just measure oxygen consumption rates across the same perturbations, and I see no difference across all the perturbations. So meaning indeed the oxygen consumption rate or the metabolic or the electron transport chain flux is not impacted. Okay, so another thing we can do is all the demand. Okay, so since it's not supply, what about demand? So demand perturbation, how do you do that? Okay, I did a lot. Okay, so this is a very crowded figure, but I'll walk you through a bit. So if you have questions, definitely ask me. For example, I can change, okay, let's see. Okay, maybe we can start with a little bit of the things that we already talked about before. I can actually disassemble spindle. There's actually a spindle, as we remember. So you can put some nacotazole, which is basically a pharmaceutical reagent that disassembles the spindle. So spindle will be gone. Okay, that supposedly you get rid of the demand from spindle, right? Because that's gone. And then you can also inhibit the actin. Okay, the actin is gone. And you can inhibit protein synthesis. So by targeting the ribosome, I think in this case, by targeting translation. So, and you can also play with iron pumping, right? For example, a very important iron pumping process is that the sodium-potassium pump at the cell plasma membrane, that is just pumping sodium and potassium in and out of the cell to maintain to maintain osmotic pressure, for example. Yes. Question?
Yes, I had a question from the from the pyruvate experiment. So, like, how exactly is it being provided? Can you explain?
Yeah. So my oocytes actually is living in a droplet. So chemically well-defined droplets. So that's also a cool thing that you can do with oocytes. That this droplet, I supply the nutrients quantitatively that I want. Right? I know exactly what's inside the droplet. The droplet has pyruvate in there, right? So therefore, I basically just titrate the pyruvate. I make the media. So I actually make the exact chemically well-defined media for the oocytes, and I decide how much pyruvate I want to put in there, right? So that's how basically, yeah.
So I was just wondering, So the oocyte is intact, right? It's not permeabilized or anything. So does uptake, how does uptake scale with supply?
Good question. Yeah, so unfortunately, I'm not measuring the uptake rate directly. I think there's some work in the literature showing that I don't exactly know the functional forms, so I do not know. But what I know is that when I increase the pyruvate, I am increasing the concentration of NADH inside the mitochondria. You can see that, for example, right from like here, you can see this is the 18.1 micromolar pyruvate, and if I increase to be 1.81 millimolar, it goes in this direction, and that is basically increasing NADH concentration. So I do not know about how exactly I'm changing the uptake rate, but I know I did change the NADH concentration. So presumably I did change the uptake rate, but I don't know like by how much.
Yeah, I understand. Just one thing, that between the first two orders of magnitude, there's not really a change in NADH concentrations. Am I right in that?
From this, it looks like that. Yeah.
Yeah, it's only when there's a 2 order of magnitude increase, that's when. Okay.
Yeah, exactly. Yeah, good question. Cool. So demand, like I said, and also I can also change the potassium levels, and that would presumably change the sodium potassium pump. And also you can also inhibit this pump with the inhibitor. So basically I changed cytoskeleton activities. I changed the translation of protein synthesis, and I changed the ion pumping. So that's what I did here. Okay, and then the data is a lot of data, but again, you can just see they are along the straight line, which is very surprising, but they are spread out across X and Y axis. So same conclusion, perturbing demand changes the turnover rate of our NADH, which is the variation along Y-axis, and the concentration of NADH, which is the variation along X-axis, but not the predicted flux because they lie. All the data points lie along this straight line. And also you do the same test where you can actually measure oxygen consumption rate under the same perturbations. Again, using this respirometer I just described, which also there's no significant differences. So it confirmed that even perturbing demand through so many different approaches, you're not changing the flux, even though you changed NADH concentrations a lot, but the turnover rate seems to compensate it for that. Okay, so, okay, any questions on this? I would say this is a lot of information. I guess still digesting, or is all clear? Okay, good. So, but okay, so here, of course, one can ask, did you actually perturb ATP levels? Okay, so I showed you, right? We can use the ATP Fred sensor to measure ATP level, right? So this is kind of application of that. But I have to say, it's very preliminary data. So therefore, more experiments needs to be done to really test this. So take this with really a grain of salt, where I only literally tested only one condition, which is nacotazole. That means where I disassemble my spindle. So if I do that, it appears that upon calibration also of this sensor, that it appears that ATP level increases after I inhibit spindle, or after I inhibit microtubule polymerization, which disassembled spindle. So therefore, I think I'm tentatively saying perturbing demand impacts ATP levels, but not the predicted or measured flux. Okay. So therefore, I'm kind of making a conclusion here, where I tested supply and demand to see if they are the factors that sets or control the oxygen consumption rate of the oocytes by perturbing systematic perturbations and measure oxygen consumption rate. The answer is no, right? So supply and demand impact NADH redox reactions and concentrations and potentially cellular ATP levels, but not the oxygen consumption rate, which is very surprising. And we term this flux homeostasis, right? So this is our observation. And okay, but the question still remains, then what controls mitochondrial metabolic flux? Okay, so, well, okay, yeah, question?
Xingbo, I had a question.
Yeah.
So when you added cortisol, and so that means that the energy consumption from the spindle is not now gone, but ATP has reached a new steady state from that bar graph. So, but the spindle was continuously consuming energy. So there was like a flux of ATP that was going through there. But if you cut down that flux, that means now ATP should continuously keep increasing, right? Isn't that true?
Yeah, okay, but that's a good question. So I think if they reach a new steady state, okay, we have to double check that. So that therefore it's a grain of salt. We don't even know if it actually reaches another new steady state. But so that's why we need to do more experiments. But let's say if they did reach a new steady state, that probably means the fluxes is redistributed, right? Some other processes that okay, either okay, no, I would say maybe two possibilities. So one is it's redistributed, some other processes that just takes over. And okay, yeah, I think that's one. And the other I think is probably the other is for example, maybe the production rate adjusted, the ATP production rate adjusted, for example. Okay, of course, here I'm thinking about this because we just say there's a flux homeostasis, but the flux homeostasis is oxygen consumption rate. I'm not saying flux homeostasis exactly in ATP production rate because I'm not measuring that. So, you know, that's why it could still be, right? The flux production rate is adapting. Okay, so what is that we have not perturbed? Okay, so we have not perturbed something just inside of the mitochondria, right? Which, for example, it's ATP synthesis, right? OXPHOS itself, or proton leak. Like I said, OXPHOS is not perfect. There's always some leak. Actually, leak could be very important. It could even have functional, you know, roles, which it definitely does in a lot of contexts that all been shown that proton leak have very, very important physiological roles. Play a very important physiological roles. So therefore, always remember there are, you know, ATP synthesis and there's also proton leak. That's how you dissipate your proton gradient. So if we perturb these two components, again, I have my reference, which is control state, the control condition, and then I can actually modulate the leak. So I can actually add some called uncoupler. This FCCP. What it does, I think it's some recent studies shows this is acting on the uncoupling proteins. So there's some uncoupling proteins, some other channels that in the mitochondrial inner membrane where protons just go through, but without making ATP. So it kind of, you know, acts on that, and then you enhance the leak. And also you can inhibit the ATP synthesis, right? And then this particular pharmaceutical reagent is blocking ATP synthesis. So if you do that, you can actually either make the mitochondria run hotter, so that basically you have a higher oxygen consumption rate if you increase the leak, or you can make it, you know, the oxygen consumption rate go lower if you block ATP synthesis. So therefore, we say the intrinsic rates of mitochondrial respiration that control the mitochondrial metabolic flux. So therefore, just to recapitulate, that we ruled out supply and demand as the controller for the oxygen consumption rate in oocytes, and we call it flux homeostasis. And we discovered that it's the oxygen consumption rate is controlled by intrinsic rates of mitochondrial respiration, like leak and ATP synthesis. Okay, so
Xingbo, I had a question.
I think, yeah.
Yeah, so I'm sorry, I probably missed out on this. Let's say when you are perturbing demand, okay, have you done that for different supplies, like supply level 1, and then you look at different demands, and then supply level 2 and different demands? Like, have you done that? Because I think I missed out on that.
I have not done that, but that's a super, super good question. I only done the demand at the control condition.
Right, because it could depend on, you know, what is your supply condition, right? So both of them could be, you know, this homeostasis that you are talking about, if that is based on independent variation, then I think you should also look at perturbing both of them together. Right?
Yeah. Actually, that's on my schedule now. So yes.
Okay.
Thanks for the suggestion. Yes. Cool. Okay, so I have about 4 minutes. I would also not go into too much details. I think I would just but give you like a teaser, probably, right? Like show you an interesting phenomenology so that you guys can brainstorm, think about it. So, like I just showed that this flux, measurement technique that I described, this is based on microscopy, right? So it can get, optimally even single pixel resolution, right? So that means we can actually look inside of the cell to ask questions: are there any spatial patterns of mitochondrial oxygen consumption rate throughout the cell? Okay, I did just that. So where I just look at again, it's a cross section of the oocytes. I just grouped mitochondria into different regions. In this case, as a distance to the membrane or to the center of the cell, depends on how you look at it. And then I basically perform this flux measurements through the, assisted by this model. This is the results. Okay, so I predict that the oxygen consumption rate, in this case, is higher towards the cell membrane, and it's lower towards the cell center, right? So therefore, there is actually an intracellular gradient of mitochondrial metabolic flux, precisely electron transport chain flux, or OCR, that exists in the oocytes. So therefore, mitochondrial OCR is not just a homogeneous quantity, right? There's actual spatial pattern. So then the idea is like, what causes this gradient? So maybe I will stop here, right? So it's open question here. So if you want to know a little bit like what experiments we've done to try to solve this, you can go into our publication. So this is being published. So we have some work down there, but it's still an open question that is actually ongoing in my lab right now, where I'm trying to figure this out. Okay, so with that being said, I'm gonna skip the next few slides. I want to first quickly make acknowledgements, right? So first, I would like to thank Needleman Lab at Harvard University. That's where I did my postdoc research, and which is also where this research is being done. Most of this research is being done at Needleman Lab, and with all the great lab members. And Yash, thanks a lot. And also for helping me out with this amazing winter school. And also Yash and I are still collaborating. And as I also talk a little bit about this spindle-associated metabolic gradient, we'll want to figure that out. And here, it looks like there's another gradient, which is radially. And we're trying to figure that out as well. Okay, so also now my lab is at Physics of Life, which is a new Biophysics Institute here at Dresden, in Germany. And I've joined quite recently this year in February. So my lab is very young, but luckily I already have some great, amazing lab members that are working with me and also collaborators. So I would like to thank them. So finally, allow me to give a little advertisement for our institute, because it's a relatively new institute. It's called Physics of Life. It's funded directly by German Research Foundation. And we have around 15 faculty members working on biological self-organization across scales, from molecular level to cell level to tissue level. And we are interested in mechanics, energetics, among many other things. So we have master program for master students, we have PhD program, and we have great postdoc programs as well. So you're welcome to join us. With that being said, finally, right, I think Yash will lead the TA session to give you some hands-on experience on the quantitative image analysis of spatial metabolism, which, for example, have been shown is very useful. For example, as example in this research, right? And there can be, given the techniques we described, I think now is a great time to dive into this and to see how we can use those great tools to solve, spatial temporal metabolism questions. So with that being said, most importantly, thank you all, and thank the organizers for inviting me. And I enjoy the school greatly, especially the questions. So if you have any more questions, I can still answer some, I think.
I have a question. So
yeah,
I don't know in what extent we can answer that, but because of the challenging way, like functionality is hard to relate to morphology in mitochondria, I guess. And so when you are changing the balance between uptake and supply and demand, there is change in NADP and with your flux, but then the oxygen consumption is not affected. But do you think that it could be like some parallel mechanisms more related to the morphology that could balance this change at the end?
Yeah, very, very good question. So morphology, yes, exactly. I think what I have not talked about, right, you have mitochondrial dynamics, right? The people know that. For example, okay, here is a comment, right? People know the mitochondria do like fusion fission dynamics, right? And that's potentially related to mitochondrial metabolic activity as well. Here in the oocytes, at least on the control condition, what we did is we did do some electron microscopy. I'm not showing the data here, but if you look at the electron microscopy data, that's how you study morphology, right? And then you can actually see mitochondria, in this case, they're not doing much of fusion or fission. They are actually pretty round, like little round spheres. And they are, they can be in contact with each other. They're like close packing and have clusters here and there, but you don't see them do much of this fusion fission thing. So they're kind of more roundish, individualized mitochondria. And some studies say that at this stage, they're like kind of immature. The mitochondria is a little bit immature. And as you develop, when you go to blastocyst stage, they start to become elongated, more like your textbook, type of morphology of mitochondria. But I have not seen the morphology under different conditions. So we've seen under control conditions, but yes, if I do demand, I do supply perturbations, there's still a possibility. Maybe the morphology could change. And I think that we need to use the electron microscopy to study that.
Okay, if there are no more questions, then Xingbo, thank you so much for this lecture and all the lectures in the past. We'll see you.
Yeah. Okay. All right. I'll see you guys.